We'll talk today about the effect of bacterial infection on TSC extracellular vesicles. Now, the Hardy Lab studies bacterial infections that affect children. And we use animal models to image this in live animals over time. Multimodal approach using many different imaging modalities that have been designed to work in mice so that we can image these infections over time non-invasively. The major one that we use is bioluminescence imaging, in vivo bioluminescence imaging. This is the bacterial are given the lux operon by recombinant DNA. So the lux operon encodes for the emission of light. So the light can be detected through the tissues of a live animal over time, non-invasively, in mice, for instance, using this instrument here, which is the IVIS instrument, the in vivo imaging system. So it detects the light from the bacteria. So you get a picture here, then the camera is turned off and the light is collected by a CCD camera. So we can watch the infection over time non-invasively. The organism we're going to talk about today is Listeria monocytogenes. This is an organism that preferentially infects pregnant women. It causes stillbirth and also bacterial meningitis and some other serious diseases in the neonate. And in the general population, this is has a high fatality rate of 20 to 40%. It's a foodborne illness, so it's acquired from contaminated food. And it's of large importance to the food manufacturing industry. If you find Listeria in your food processing, you have to recall the food because of this high fatality rate. Many food recalls due to this organism. So our prenatal infection model, Listeria infects the placenta of pregnant women. This is how it crosses to the fetus sometimes, and also causes the abortion and stillbirth. Here you can see this mouse here, pregnant mouse infected with Listeria. These are the infected placentas. And then we also use multimodal imaging to look at other aspects of this, ultrasound to measure fetal heart rates, MRI to measure or to look at their morphology, and then it can be transmitted to the fetus. And we can also look at what happens to the fetus after it's born. So this type of infection can affect fetal brain morphology and behavior of the fetuses after they grow up into adults. So here's what we're going to talk about here. The Listeria infects the extracellular or extravillous trophoblast. So that's these cells here in the fetal villi. And so let's look at that a little bit closer here. Listeria is an intracellular pathogen, so it comes with the maternal blood inside cells, and those spread from cell to cell. So it can infect the extravillous trophoblast, and then it gains access to the villi. It cannot get through syncytial trophoblast. It cannot infect through that layer. That's a barrier for all kinds of things, especially bacterial infection. But it can get access to the syncytium through these extravillous trophoblasts, and then it can cross this basement membrane and get access to the fetus. And crossing the basement membrane requires a special protein from Listeria. Here's a bioluminescence image of the infected placenta and this fetus here. You can see this infection. We can image this to high resolution in excised animal. And then we've also done studies, I'll talk about in a minute, of the transcriptome of the placenta, the metabolites of the placenta, and also the transcriptome of the fetal brain in response to placental infection. So here is this crossing that I was talking about. The protein that is responsible is called internalin P. It's a very, very interesting protein of Listeria. Here on the right, you see this fetal mouse is infected in the placenta only, but this one, it has crossed over into the fetus, and that requires this protein, internalin P, to cross that basement membrane and get access to the fetus. You see this kind of creepy image here of the eye, and that's because it has Listeria has gone into the blood of the fetus, and the eye is very close to the surface. The blood has very little tissue in between that and the camera, so you get this lit up eye that looks kind of scary. Okay, here's we've studied I talked about the transcriptome. So here's mouse infected with Listeria in the placenta. We can compare this placenta that is infected versus that one that's uninfected, and we can get all kinds of information in the same animal. So a control study from the same animal. We see that many things that affect the birth process and brain development are perturbed by infections. So lipoxins, prostaglandins, other kinds of things like this are upregulated and downregulated by this infection. And we've done a bunch of analysis on that, my grad student, Kayla. So here you see in the transcriptome, we see these enzymes upregulated that are in the eicosanoid pathway, and they're responsible for the upregulation of these hormones. And those, some of them are used in humans to induce labor. So that's one of the reasons we get preterm birth with these mice when they're infected. Now, in humans, we'll get now to extracellular vesicles. In humans during pregnancy, the EVs go way up in maternal blood. So this is a paper showing the increase in extracellular vesicles during pregnancy in humans. And so these extracellular vesicles, a great number of them are made by trophoblasts. So they regulate the immune response. For instance, if a woman has preeclampsia, you can take her extracellular vesicles, inject it into a pregnant mouse, and that mouse will get preeclampsia. So these are very important in the immune regulation of the maternal fetal interface. So the cells we use to model that are these murine trophoblast stem cells, mouse trophoblast stem cells. They grow in a colony like this here. You know, they grow in these colonies with these growth factors, fibroblast growth factor, activin, and you have to add heparin to these because there's a lot of serum in there. And so they grow like this. You can keep them growing for many passages using these growth factors. In mouse cells, not in human, I don't believe, but in mouse cells, if you remove the growth factors, they spontaneously differentiate into this giant cell. See that? These giant cells form spontaneously when you remove the growth factors. And you can also induce them to differentiate in using other methods. So John Koletka in the lab infected these cells with Listeria. That's the target cell of Listeria. Remember the extravillous trophoblast. You can tell that we got intracellular infection by these actin tails here that Listeria forms. That's how their bacteria spread from cell to cell. So these are one of those colonies infected with Listeria. And so he differentiated the trophoblasts, the TSCs. He differentiated them using a couple of different methods, including just removing the growth factors. And it inhibited the growth of Listeria, which is kind of what we expected, because the trophoblast that's infected is that extravillous one and not the syncytial trophoblast or other forms that trophoblasts take. So then he made EVs from the infected and uninfected TSCs. So here's just the method. I think everybody's familiar with construction or purification of extracellular vesicles. You get the large ones and the small ones, and of course the nomenclature is changing over the years. These used to be called exosomes, and now they just call them large and small EVs. At least that's what I'm told. Anyway, I think it's still up in the air. But anyway, so these large EVs are affected in size by Listeria infection, but really not the small ones. The large EVs, they say, are many of them are composed by microvesicles, which bud out from the cytoplasmic membrane rather than being produced from an internal process the way exosomes are. So these got perturbed probably by actin effect of the actin cytoskeleton by Listeria. We don't know that for sure. Okay, we take these EVs and we use them to treat macrophage cells to look and see if they alter the responses of these macrophages, and indeed they do. So here is these are raw 264 macrophages, mouse macrophages. you treat them with the regular EVs from the uninfected and the infected, and you see that you get an increase in tumor necrosis factor, TNF. So that is increased by the treatment of these. There's another cell line, the J774 macrophage, large increase in TNF when they're treated with the ones from the infected TSCs, not the uninfected ones, right? So infection is altering the function of these EVs. It also alters their target cell susceptibility to Listeria. So here's the J774 cells over here that you see no change when you treat them with any of the EVs. That's a permissive cell for Listeria infection. But to our surprise, we got an increase in susceptibility of raw cells when we treated with EVs from infected TSCs, not from uninfected, not so much from uninfected. And the same thing in bone marrow-derived macrophages and also in TSCs themselves, although Listeria grows very slowly in TSCs, and so we didn't see too much of a difference there. So a surprise in the field of Listeria to get an increase in listerial growth by treating these cells in this way. So very interesting. Now we looked at the protein content of these. This is PEG-10 was upregulated in the TSCs from infected or EVs from infected TSCs. PEG-10 is a TSC differentiation factor. But the main thing we saw were many of these ribosomal proteins and also nuclear proteins, and those are a sign of damage-associated molecular pattern, so DAMPs. So when cells are damaged or perturbed in some different ways, you get proteins appearing in the cytosol that shouldn't be there, including ribosome proteins that come off of the ER, including histones and other kinds of things, and those can be loaded into EVs, and then those EVs can affect target cell by their content containing these DAMPs, damage-associated molecular pattern. Now, one thing we did not see in these EVs were any proteins from Listeria. There were no bacterial products in these EVs at all when they came from an infected cell. So that was very, very interesting to us. So because that's not the case with macrophages. Macrophages, you feed them bacteria and they package the bacterial products in the EVs. And that's how they can transmit things like immune stimulation. Not the case with these TSCs. There was not a single bacterial protein in any of the TSC samples that we looked at. So the entire process of the modulation of the immune response was due to these damage-associated molecular pattern. And so that we're very, very interested in, because we believe that if the placenta is being perturbed or in some way altered, that these DAMPs could be a signal to the maternal immune system. And so we're very, very interested in looking at these. And I urge anyone who is here who's working with EVs from TSCs to look for DAMPs, because that might be a signal of that EVs are used to communicate with other cells. We decided to look at the RNAs in these EVs, and we found a lot of ribosomal RNAs. So that was another DAMP signal. This is a known thing, these DAMP signals, a ribosomal RNA appearing in the EVs, okay? And this is just a sort of interaction map, sort of, interaction of these pathways showing here's PEG-10 and these ribosomal proteins here. And all of these are signals of including histones and tubulin and things like that shouldn't be in the EV that show up there when it has been infected. So this is one of the communication methods, we believe, during infection of the placenta. So very, to me, we really are interested in this. Then we take these EVs and inject them into mice and infect them with Listeria. So what happens? Now, we got a reduction in infection with uninfected EVs and a change in the infected ones as well, but this is just a preliminary experiment. Really, these were not statistically different samples, even though they might look like it here. This is the spleen, by the way. Listeria infects the spleen in non-pregnant mice. So here, we really want to pursue this more. We would like to do this in pregnant mice. We would like to see what these EVs are doing. We find it very interesting that they increase the level of infection in some cells and they turn on TNF. That may be what's responsible for this reduction here. The induction of TNF may reduce the infection levels, but we really have a lot of work to do here. And we're very excited about this, but very preliminary data that John did. So here's the TSCs that Accelerated sent to us. Thank you guys for doing that. I mean, that was just a really cool thing. Anyway, so they're looking really nice. They look way different than the mouse ones. And so I think we're very excited about this and interested in growing these. So we just plated them out on the mesenchymal medium here and so they look very nice. We tried coating and uncoating the substrate, the flask. The coating really helped a lot for them when we split them. And so we've been looking at a bunch of different growth properties in these. And here's the Captivate platelet lysate in MEM. They grew really nicely in that medium. And so Pramika and I, my student and I are exploring how to grow these. We're going to get used to growing them, and then we're going to do our Listerian experiments with them to see if they express DAMPs the same way the mouse one did. And so we're very interested in that. So here's the summary of sort of our TSC growing experiments. In the mesenchymal medium, the cells didn't grow very fast, but they had really good morphology. This medium is also clear. It doesn't have any serum in it, so that's very nice. And there's no animal products in it, I don't believe. And so so that's going to be suitable for making EVs. So that's the medium we're going to use when we do our infections and make our EVs and characterize them. But the MEM platelet lysate medium, the cells grow very well in that medium. They grow really nicely. The morphology is good, but that medium is full of junk. I mean, you look in there, it looks like it's contaminated or something. It's not, but it's just full of all these, I guess, platelet. It looks like sonicated human blood or something. But anyway, they grow really nicely in there. We found out the hard way that it requires heparin. Yeah, actually, my student Pramika looked up the stuff on that. So without heparin, it will gel on you. Anyway, so this is a human product. We have to keep that in mind. But this we're going to use for our differentiation. And so it's nice to have these two kinds of medium. And I've heard from Jen that you guys have switched to another one now. And so we'll start growing that. But we're just really happy. The cells are growing nicely. They look great. And we're ready to do our experiments. So the future plans, we're going to infect these and characterize the EVs. We're going to look for these DAMP signals because I think this is going to be important. And I really think this is going to be important for not only just for our case, but for maternal immune responses in general. I think this is going to be an important thing, these DAMPs. So I really am hoping people will look at them. Then we're going to differentiate them, try to differentiate them with different methods. Here's this CHUR that we use for the mouse one, and maybe retinoic acid or some other ways to get them to form syncytia. So I'd like to thank many people who worked on this, Kayla and Justin and John, my grad students, and also especially Pramika, who's growing the human TSCs with me, and she's done a great job. And so thanks very much, everybody.